We started it. All right, I think we started. Okay, so welcome. I'm going to share my screen and go over to here, share. There we go. All right, so a couple of things that we need to get out of the way before I start on this lecture. I think I just sent out my first email on Avenue to all of you. I didn't receive any sort of confirmation from Avenue alerting me that that email went out, but I did compose an email and I did go through a process where it appears to have been sent. So please alert me if you receive that email, one of you or 60 of you, it doesn't matter. Just somebody let me know that that process worked, and so I don't continuously think that I'm corresponding with you, and in fact, I'm not. All right, so let's see. I'm gonna make sure that I don't go over too far over time for this lecture. Okay, so the other thing that I need to say to you is that, I scheduled the first two exams. So one of the participants in the class articulated that she would prefer that I schedule the dates for the exams in a firm capacity so that you guys can, schedule your lives and make plans based on those dates. So the two dates, in case you haven't received the email, or in addition to that email, just to reiterate it, are October 8th, which is a Thursday, and November 12th, which is also a Thursday. And so about a month apart, some change, and about a month until the first exam. A couple of apologies. Number one, my neighbors immediately outside of this room are loud. And two, the other thing is I have a bunch of black-faced conures outside of my window that are feral here in Tampa. My parents are quite ill, so I'm here taking care of them. I'm also not a Canadian national, and I'm not a permanent resident yet, and so the Canadian border is closed to people who are not essential workers, and I am viewed not to be an essential worker. So I'm actually trapped in Florida, and there are feral black-faced conures outside making a lot of racket. And because I updated my webcam and have a nice little sound, what do you call it, microphone in front of me, you're going to be able to hear some of those black-faced conures. They would annoy me as a student, but I apologize. I have no mechanism of making those birds go away. That doesn't render me likely to be arrested. Anyway, so October 8th, October 12th, please let me know if the Avenue thing goes through. It's important that that begins to work, or it is, that I get a sense that it is working. Anyway, enough distractions. All right, so this will be the at least second quarter of this lecture today, and then possibly we will do the third quarter today. We'll see where we go. I mean, it's quite likely that I will end up running behind in these first few lectures, and then we'll play catch up in units 3 and 4 because they're more concise. All right. How does evolution work? So we already talked about how ecological social interactions are categorized in terms of their costs and benefits to donor and recipient, 4 general categories and examples of each, and prototypical examples of each. And now we're going to think about, just quickly reviewing how evolution works and how the costs and benefits of social interactions aggregate together to influence the evolvability of social traits, which could be things like cooperation, it could be altruism, it could be selfishness. All of these things are kinds of social interactions. And so how does evolution act based on those costs and benefits in various currencies in aggregate together to foster whether or not a trait that's novel, a social trait that's novel, spreads in a population, or whether or not it stagnates, withers, and dies, and doesn't get off the ground. All right, so evolution. It's defined as change in the heritable characteristics of biological populations over successive generations, right? So over time in the same population, there will be traits that are transmitted with fidelity from parent to offspring, like height, or eye color, or hair color, or aggressiveness, or sociability, or coloration on individuals' back, and that these variations among individuals will tend to be transmitted with some level of fidelity from parent to offspring. So tall parents produce tall offspring. Tall parents have tall offspring. Short parents have short offspring. Aggressive parents have aggressive offspring. Docile parents have docile offspring. And that if some of these traits are linked with enhanced performance, either in survival, or reproductive output, or mating efficiency, or whatever, that those will tend to be overrepresented in the next population, in the next generation, and that will occur over many, many generations. And through time, you will see shifts in the relative frequency frequencies of various kinds of traits in the population. The mean will switch back and forth, or the frequencies will shift back and forth if we're referring to relatively discrete traits, like it's either this one or it's that one. But most of the traits that we're talking about for social evolution are going to be continuous traits that have many genes that influence them. So we're going to be watching shifts in the mean through time as a consequence of the selective environment that they're exposed to. So it has to be heritable for there to be an effective response to selection. Heritable means that it's transmitted from parent to offspring. If tall parents, when we take a dam and a sire, a female and a male, and they're both tall, we breed them together, and they have a random mix of offspring that have no significant statistical resemblance to the height of their parents, then it doesn't matter whether it's good to be tall or short or dark colored or light colored or aggressive or docile or whatever, because even though there's ongoing selection favoring those traits, they're gonna have total slurs of offspring in terms of traits that could be any possible combination, and which means that it's going to be reset generation after generation after generation. So there are lots of traits that have a low level of heritability. There are lots of traits that have a high level of heritability, which means they either have a low level of transmission of fidelity, so tall parents can produce a variety of offspring, although they might slightly tend to be tall, or a high level of heritability would be that two tall parents produce almost strictly tall offspring. And this heritability turns out to be important, whether it's high fidelity, or low fidelity, because it basically says whether or not, how efficient the response to selection will be. So if there's a very high level of heritability and there's strong selection, then we'll see fixation of that trait that's favored very swiftly, because there'll be very little noise in the traits of the offspring relative to the traits of the parents. If the heritability is low, then even very strong selection will have a relatively weak response through time, because even though there might be a strong advantage to being highly aggressive, their offspring of those aggressive parents are going to have a variety of different kinds of traits. It's going to be quite messy, and it'll take a long time for traits like aggressiveness to proceed to fixation, if they ever do. So, natural selection. Of course, I've got a picture of an old person here. You know, Charles Darwin. How can you talk about how can you talk about evolution without natural selection? Natural selection is just one mechanism of evolution. It is not the only mechanism of evolution. It's the one that people think of immediately, but it is almost certainly going to be one of your exam questions as to whether or not there are different kinds of evolution or mechanisms of driving phenotypic change across generations that are heritable, apart from natural selection. I will be hoping that I'm not hoping, but I will be gunning for people who assume or fall back on their presuppositions that natural selection is the only kind of evolution. That is not true, but it is probably one of the more efficient and pervasive kinds of mechanisms of evolution. So we review it because it's important, and then I'll contrast it to other kinds of evolution, common mechanisms of evolution, in just a second. Natural selection, the differential survival and reproduction of individuals due to differences in phenotype. So it's deterministic, it's non-random. Some traits do better than others, they increase in frequency, and that will change the average phenotype and trait of a population through time. When the traits that are favored in an environment differ across populations, this will tend to cause reproductive barriers to evolve between them to make sure that you don't get suboptimal mixing of adaptations between populations that have local adaptation. Local adaptation is just the idea that the traits that are favored vary by their environment. And so if different populations of the same organism occupy very different kinds of environments, those could favor different kinds of traits in the individuals that live there. And through time, we'll see the average trait value of those two different populations diverge. So we'll talk about local adaptation probably a little bit more later on, but it's important to note that once you have local adaptation, so natural selection generally also favors reproductive barriers between those populations to prevent traits that are evolved to be adapted for one population from mistakenly adulterating the adaptations of a second population when there are migrants. And that's how new species are thought to primarily evolve. dispelling misconceptions. I said it before, I'm going to say it again, I'm going to say it in writing, I'm going to read it aloud to you. I'm going to tell you what I'm going to tell you, I'm going to tell you it, and I'm going to tell you what I told you. All right, so not all evolution is caused by natural selection. You probably already know this, but just again, like, it bears repeating because so many people just equate natural selection because it is a highly efficient and it's the most famous form of evolution. It is not the only form of evolution. And the two most common sort of demographic or chance event type mechanisms that also drive evolution that you'll hear about in intro biology and probably iteratively in other biological courses are genetic drift and bottlenecks. So genetic drift, I'm going to read it aloud to you. Then we'll go over each one just briefly with thematic examples. Genetic drift is just variation in the relative frequencies of different kinds of genotypes, and typically in a small population, owing to chance disappearance of genes as individuals live or die or fail to reproduce. So it's just noise. It's just the randomness of life that causes fluctuations in the average genotypic composition, and therefore the trait composition of the population through time. Bottlenecks, I don't even like to even think about bottlenecks as being all that thematically different from genetic drift, but I'll read you the definition, and there's a fuzzy barrier between them. Bottlenecks are a sharp reduction in the size of a population due to environmental events such as famines, earthquakes, floods, fires, disease, dispersal, etc. So that is, there once was a large population that had lots of individual animals inside of it, reproducing with each other, competing with each other, caring for their offspring, etc., etc. And then something bad, or at least extreme, happened and took many individuals and brought it down to very few individuals. And when you do that, that means that whatever traits those individuals that just managed to survive, whatever they have going for them, that's what's available for natural selection in the time that follows. And so those extreme events oftentimes cull lots of the diversity from a population, and that means that the new population, at least for a time, the population that persists through the bottleneck will tend to be more homogeneous afterward, because whatever traits those individual survivors had, that's very likely to be the milieu of traits that population can exhibit for the next several generations, or maybe even dozens or hundreds of generations. All right. Genetic drift. Oh my God, there's a shoe accidentally stepping on these beetles. So imagine a population, a fictitious and pixelated population of beetles. I say pixelated because, well, whatever, it is pixelated. I know it's pixelated. Anyway, imagine a fictitious population of these beetles where there are phenotypes of green beetles and these tan beetles, and that they're transmitted with fidelity. Green parents produce green offspring, cream or tan-colored beetles produce tan-colored offspring, blah, 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 blah. Strict inheritance, blah, 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 blah. All right, just by chance event, you might have a mortality event where a subset of the population just die. A disease goes through, and in this case, a shoe steps on some of them. A hurricane goes through. A volcano goes off. Or just chance, by chance, the green ones get more mates in one generation, or the cream ones get more mates in a generation. And it has nothing to do with the fact that those traits confer an advantage, but just by chance alone, like lottery events, you can get fluctuations in the survival or reproductive output that have nothing to do with their traits, but just are anomalous, right? And so after those anomalous events happen, you'll have a shift in the relative representation of one trait versus another. Genetic drift is most powerful and most influential in small populations. If you have very few individuals, chance events change the relative frequencies of those different traits much more dramatically. If we had an enormous population of beetles, hundreds of thousands of them, a foot stepping on two of them is not going to matter, because two individuals relative to a population of hundreds of thousands is a drop of spit in the bucket, versus if we have a population of 9 beetles in this picture, and 2 of them die, well, that's more than 20% of the population. So chance events are more influential in changing the relative frequencies of various genotypes and therefore phenotypes in small populations than large populations. And following from that, bottlenecks are really influential because they take giant populations and they basically winnow it down to just a small number of individuals that manage to live through whatever catastrophe has befallen this population. So, looking at this little literal I guess it's the literal bottleneck example, you have a bunch of these little tiny different colored balls, right? And then there were many of them, but then there was a bottleneck event, presumably these are some sort of like model organism that perish because it's a volcanic event or a natural disaster, or there's a neighborhood that now goes up that turns the former woodlot into a bunch of lawns, and that could kill lots of individuals of a focal species. And then the small number of survivors are oftentimes just a random subset of the original traits that were in that population. But whatever traits those survivors have, that is just what's available for the next many generations. And so those will proliferate again, and the population will grow in number, moving from this figure from the middle panels to the right panel, to the right side of the panel. And if they have no selective advantage, whatever the chance surviving phenotypes were, their representation should be overrepresented in the new population relative to the representation in the former population. So in this example, there were a lot of little yellow balls, like some sort of yellow organism, and then a catastrophe happened, and just by chance, very few of those yellow individuals managed to make it through the culling event. But then afterward, everyone reproduced again in a neutral way where everyone had the same reproductive output. And now in the new population, the representation of that yellow phenotype in this right-hand bottle is just much lower than it was before. So it's like genetic drift, but it's an extreme version of genetic drift where a chance event takes a huge population, winnows it down to a small population. And that also means that at this like small population size in the middle here, other genetic drift elements, other chance events, can really have a large influence on these populations. So the mixture of extreme bottlenecks, big populations to small populations, and then drift, that statistical noise of chance events in small populations combine to be very influential over evolutionary time. So we have no idea what proportion of the genetic change that we've seen through animal evolution is driven by drift and bottlenecks. But it's at least in some contexts, things like island biogeography, when new islands are colonized by individuals, we think bottlenecks are probably incredibly influential. Here's an Anolis lizard, an adorable Anolis lizard, a male. I don't know why, but I guess it was just more colorful. But it does not just, chance events like bottlenecks are not just, in theory, a bottle of mixed phenotypes, and then they go through, some reduction in population size and they grow again, but they happen all the time with real organisms. They can be episodic events that happen semi-regularly. So Anolis lizards that happen to occur on tiny Caribbean islands might have populations of hundreds of Anolis lizards, but if they have very little structure to hide in during these catastrophic storm events, which can happen every few years, every couple of years, every 5 years, every 10 years, 15 years. One of these giant storms can cross over these tiny little Caribbean islands filled with these lizards and eliminate vast quantities of the population. A population can go from hundreds and hundreds of lizards to just a few dozen. And these constitute semi-regular bottlenecks that these populations go through. And if bottlenecks are happening all the time, then it means in these populations, chance events, rare events, are perhaps responsible for a large portion of the traits that we see in that population now, the fixation of certain kinds of phenotypes. As I mentioned before, I'll mention it again now, population size is very influential in determining the relative influence of bottlenecks and genetic drift on phenotypic change. So just like if I talked about a population of 9 beetles versus 100,000 beetles, if we talk about a small group of hunter-gatherers that have like a few dozen individuals, like this indigenous tribe in Kenya, versus Nairobi on the right-hand side, where there are many, many hundreds of thousands, millions of people in Nairobi, I guess it's maybe only about 1 million people in Nairobi. I should probably check that out. But anyway, there are many more people in Nairobi total than in a small hunter-gatherer tribe. Precise numbers don't really matter in this case. So if something like a novel virus were to go through a population and kill just a small number of individuals, we'll say 10 a family unit. If 10 individuals died in a family unit in a small hunter-gatherer tribe, that could constitute a large representation, genetic representation, or demographic representation of that total social unit, which means small chance events that affect a small number of individuals will proportionally have a larger impact on a small population like a hunter-gatherer tribe. If 5 individuals die in Nairobi, demographically, it's almost nobody. It's almost, I mean, it's not a percent, it's not a tenth of a percent, it's an infinitesimally small number of individuals. So those chance events, 10 individuals dying, 5 individuals dying, 12 individuals dying, just by random chance, would have a large impact on the proportion of various kinds of the frequencies of different phenotypes in a small population, and a relatively negligible impact on a large population, unless a bottleneck transpires, like a very bad disease goes through on the right-hand side, and then drift would be more influential. Evolution requires heritability. I mentioned it before, I'll mention it again now. Here's this like peppered moth example where, you have moths and they have various kinds of colors, but then they're selected by predators, and the more conspicuous phenotype, these white moths here, get picked off by what are these blue jays, or maybe they're scrub jays. They look like blue jays to me. And then the relative representation in the next generation of these dark versus light phenotype is biased towards the dark phenotype because guess what? It's dark, it matches the spark, and so they're more cryptic. And so this is predator-induced selection that's favoring a darker phenotype. If by chance those surviving dark moths produced a totally random representation of both light and dark colored moths, there would be no phenotypic response to selection. Those predators could be picking off those cream colored individuals all day long. But in the next generation, guess what? Too bad for them. Like they're gonna have these dark moths are gonna produce offspring that have the same probability of being cream as being dark, and so that disadvantageous phenotype, if it has no heritability at all, it's just going to show up again and again and again and again, based on its probability of being produced by a parent. If heritability is high, though, then we should see an effective response to selection. If these blue jays are picking off all these cream-colored moths, well, then these dark-colored moths are going to tend to produce more dark-colored moths, and that cream phenotype, bad news bears for it. Its days may be sadly numbered, i.e., the dark phenotype might proceed to fixation, which means that the other trait variant is gone. Fixation, it's fixed. All right, heritability. So I'd already talked about how heritability is a metric that describes the degree of resemblance and transmission from parents to offspring in terms of their traits. This is also defined statistically as the proportion of variation in a trait explained by descent and familial relationships. It's the same thing. It's just the degree to which parents resemble, family members resemble each other, or share traits as a consequence of descent. So heritability is usually expressed as an R, a little r, and it is a statistical property that varies between 0, which means low heritability, like those offspring have a very good chance that they look nothing like their parents, and high heritability. 1 would be like perfect heritability. Whatever traits your parents have, you're getting that. Higher the R, the more efficient there's of an evolutionary response to selection. I mentioned before that if those dark-colored moths are spewing out cream-colored babies, well then it doesn't really matter how much those predators or those predator jays are picking off those cream-colored moths. That cream-colored phenotype is going to be back in no time, like back for a second inning. Anyway, so versus if there's a high heritability, that cream-colored moth would have its days numbered, right? But all traits vary between 0 and 1. There's no perfect there tends to be no perfect heritability, and almost no trait has a heritability of 0. There's usually at least some degree of transmission from parent to offspring. So basically, using heritability, you can estimate how quickly a trait that's favored will proceed to fixation if it's under selection, stable selection. So if a trait is favored and has a high level of heritability, then that means very quickly that trait will proceed to fixation. It will dominate the population. If it has a low heritability, then it'll take many generations for that more advantageous phenotype to reach high levels of representation and then fixation. So little r, although it says like the degree to which the trait is transmitted from parent to offspring or between family members, that's a statistical thing about how much they resemble each other. Parents and offspring, or aunts and uncles, or cousins, or half-sibs that share the same father but have different mothers. They share a proportion of their genome in common, and you basically use statistics to say how much of that trait variation that we see in aggressiveness, docility, sociability, whatever, is associated with those relationships, those familial relationships. it doesn't exactly mean that it's genes that's causing that resemblance, right? Lots of things run in families that aren't strictly because of your genes. You inherit quite a lot apart from your genes from your parents, like the nurture component of the nature and nurture. And those developmental effects can be transmitted with fidelity from one generation to another, and not because you have some gene that just makes you do this thing, because of the developmental environment you were reared in. So, and a high R, a high level of heritability, of course, can mean that there's something else driving that resemblance, like culture or developmental environment, that can be transmitted. And a low heritability doesn't necessarily mean that there are no genes that cause that trait. It might just mean that there's no variability in that trait. So just as a mock example, how do we estimate heritability in a trait? So Geospiza fortis. Geospiza fortis is one of the Darwin finches. There are many Darwin finches, and I'm sure you've seen them before, but here they are again. Here's an illustration of a Darwin finch. And let's pretend that we had a mating study where we took parents, dams, females, sires, males, and they have various bill depths. A thick bill depth would mean that they are able to crush lots of nuts and seeds, and then if they have a narrow bill breadth, then they can't produce so much jaw pressure, but maybe they are more gracile and able to nimbly handle small seeds, for instance. If we mate thick-billed dams to thick-billed sires, and we breed them together, in this example, they tend to produce offspring with an average bill depth that is thick, like their parents. If you take thin-billed parents, you breed them together, you tend to get thin-billed offspring. And so you get this nice positive relationship between the average bill depth of the parents, the x-axis in millimeters, and the average bill depth of the offspring, y-axis. Presto changeo. All right, but how do you actually estimate the heritability? Heritability in this instance is the slope of that line, the slope of the relationship between the average phenotype of the parents and the average phenotype of the offspring. This slope is steep. It'll never be 1, but it will be steep. And so in this case, the offspring very much resemble the parents. If that slope is shallow, then that means that even combinations of parents that have very thick bill depths are going to have relatively noisy offspring. Some of them, yes, will tend to have larger bill depths, but they'll still be producing some offspring that have narrow bill depths, and vice versa. So parents that have thin bill depths will sometimes produce individual offspring that have thick bill depths. That would happen if there was a low heritability, and that slope would be more shallow. So the slope of that relationship is the most common mechanism that people use to evaluate heritability. And in this case, the heritability is enormous. 0.9 is very, very, very strongly heritable. Most things don't have that high of a heritability, and certainly not most behavior, because there's so many environmental influences and many genes that contribute to the phenotypes that most of us are interested in this course. We're not going to study bill depths. All right. Just because there's no heritability doesn't mean that there aren't genes that influence it. Let's look at your heart. All of the people in this class right now, I'll tell you right now, I'm going to make a guess. You've all got 4-chambered hearts. All of you. You wouldn't be alive right now if you didn't have a functioning or more or less functioning 4-chambered heart. What's the heritability of the 4-chambered heart? We don't really know what its heritability is, because there's no variation in the trait. So one way that we can get no significant heritability is that there's just no variation. The trait is fixed. I use the 4-chambered heart because I'm like, haha, like, there's no variation because you can't have variation in it, as a sort of as a fun example. But the reality is that many populations don't have any heritability, because if aggressiveness was favored, all of those docile individuals have been culled from the population, and now everyone has a very tight, small variation in aggressiveness where they're very highly aggressive. And if you asked, okay, what's the heritability? There would be no positive slope in that little cloud of aggressiveness left in the population, because all of the genetic variation is gone. It doesn't mean that there's no genes conferring aggressiveness in this population. It just means that all the variability in that trait that would have allowed you to test for these familial resemblances is gone. If you drew a line through that remaining cloud, they're all so highly aggressive that it's basically a flat line. if you don't believe me, then go back to this GeoSpiza example and just eliminate all of the individuals until bill depth, I don't know, 10 millimeters, and then try to draw a line through that. You'll see that it's flat, flat, flat. It doesn't mean that there's no genes influencing those bills. There are genes influencing those bills. It's just we can't tell family resemblance any longer. And the four-chambered heart, boy howdy, we can't evaluate the heritability of the four-chambered heart in humans. And why would we want to? All right, heritability of wealth. All right, here's a great example. There's a heritability of wealth varies. Oh, I should close that parentheses. Sorry, anyway, whatever. Heritability of wealth. There are rich people, there are poor people, and there are intermediate people, there are of intermediate wealth. Blah blah blah. I don't mean to binarize. All right, so if we were to look at parent wealth and offspring wealth in various nations, you might surmise that, oh my God, there's wealthy parents tend to produce, on average, the next generation, if the parents are dead, wealthy offspring. Oh my God, impoverished parents, and we're talking about humans right now, tend to produce impoverished offspring. It's not because you have a gene that causes you to produce money. At least not, I mean, like, you probably have some genes that influence your propensity to, like, plan and sequester resources, blah, blah, blah. But, like, a lot of this is just, like, you just didn't have the same opportunities as somebody who had two wealthy parents, right? You didn't have old school money, you didn't have, like, old school wealth behind you. So, and also I should say that the degree of resemblance between parents and offspring in wealth in human societies varies enormously between societies. I think actually Canada has one of the lowest heritabilities of wealth, which means that places like Canada have enhanced probabilities of individuals changing their station in life, their resources, that they have more upward mobility because there's greater accessibility of chances and opportunities. In some portions of the world, the heritability of wealth is enormous. Like, if you're born in a hovel, you're not going to get out of it, almost certainly. At least the vast majority of individuals born in those environments will tend to transmit them to their offspring, and it's not because of not because they have a gene coding for a protein that causes them to be impoverished. So that's cultural inheritance and transmission of things like resources. And admittedly, in most animal societies, we don't have things like systemic wealth and bank accounts and 401ks and Roth IRAs and blah, 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 and stock markets. But there are things like having parents that just had a lot of resources. So when they yolk their eggs, individuals are born with more resources. They can take greater chances. And they're less susceptible to chance events like diseases. They have stronger functioning immune systems. They're more likely to acquire mates early on in life. They're more likely to be able to mature earlier and become reproductive earlier in life. And so wealth, in this case, can become advantaged in a non-human animal society, and the analog is quite strong. And my God, here's an example. We've got a fat hamster on the left-hand side here, and fat hamster mothers will tend to produce higher quality offspring. They'll produce higher quality milk for their offspring. They'll be able to imbue them with more resources. Those offspring will grow more rapidly. They'll have higher functioning immune systems. They're going to have a jump start in life relative to the emaciated mother that's barely able to reproduce this season. Those pups are going to be less well provisioned, and so they're going to start life at a disadvantage. And so, is there a gene that strictly codes for fatness that explains most of this variation, or wealth, or chance, or good fortune? No, but that doesn't mean that small differences in starting resources can be transmitted, or even large differences can be transmitted from one generation to the next. You can stop these transmission events by intervening as a researcher, or and they could be environmentally interrupted by chance. Or by fluctuating environments, but without a perturbation, quite often, lineages of animals that have advantages socially will tend to transmit them through time, and disadvantaged individuals will tend to transmit them through time, and it's not because of strict genetic inheritance, right? It can be basically transmitted environmental differences. It can also be imbued epigenetically, so like this begins to really like blur the lines between there are genes influencing your behavior, there's an environment influencing your behavior, but then environmental differences can begin to silence components of your genome and enhance the expression of other components of your genome via chromatin remodeling and other mechanisms during development that then cause the expression of that genome to change more or less irrevocably, or at least in a semi-stable way, over the rest of that individual's life. So a classic example that's probably best worked out is whether or not you have a good mother or a bad mother as a mouse. Good mother mice that are not stressed, that have lots of resources, spend a lot of their time licking their young and taking care of them. Emaciated mothers that are not doing well, that have had a high stress environment life, tend to not spend very much time licking their offspring. Mothers that lick their offspring oftentimes will tend to produce an epigenetic cascade that will change the expression of the genome of her offspring to be more relaxed adults. They're less anxious, they're less fearful, they tend to be in higher body condition as well, and those offspring, when they become reproductive themselves, the females will tend to lick their offspring as well, and tend to transmit this relaxed, nurturing state across generations. But you can interfere with this experimentally. You can just muzzle that mom. That nurturing mother that has all those, like, that tendency to lick and take care of her offspring, muzzle her. She can't do it now. Guess what her offspring do? They turn out to be neurotic. And conversely, these neurotic, anxious mothers that don't spend much time licking their offspring, that tend to produce highly anxious and low level of licking in their offspring, you can interrupt that experimentally. You can say, I know that this mother is neurotic and she's not licking his offspring. It's not causing this chromatin remodeling, methylation pattern that results in an anxious offspring. But you can just experimentally take things like a Q-tip that's warm, or a warm towel, and you can just rub those offspring, and it mimics the same effect as a mother that licks them. It's just warmth and contact that pairs together, that creates that chromatin remodeling cascade that will now intervene in those anxious lines and cause the next generation to be more relaxed, more nurturing. So these anxious, neurotic, and calm and nurturing lines that are dynastic can actually be intervened by experimentalists, and presumably are intervened by the environment every so often, under natural conditions, to cause the opposing phenotype. Not because they have a gene that strictly codes for being neurotic or nurturing, anxious or nurturing, but because they have these dynastic developmental environments that they're inheriting that change how their genome, how their molecular library is expressed. To what degree those genes can be expressed. All right, I want to make sure that I haven't gone over too far. Well, all right, so I've gone pretty far. I'll probably, let me see. Okay, I'll finish this one section, and then evidently you're just getting the second third today. I apologize. Okay. So it could also be cultural inheritance. So a classic example, which is actually worked out by a former Mac professor, Jeff Galef. Anyway, so let's see. Rats. Rats live in societies where they learn about foraging opportunities from each other, from little rat cuddle piles. And the kinds of food preferences that rats have are oftentimes copied from demonstrator individuals that have had successful foraging events in the recent past. So let's imagine these two rats in the middle here, that we allow one of these rats to be our demonstrator rat in an experiment we're conducting. And then the observer rat is stuck at home in its little cage. All right, we take the demonstrator rat, we let it come out, we let it eat rat chow, and we flavor. So let's pretend that we have lots of these pairs of rats. So we're actually running an experiment with replication. And it's a subset of these individuals. We take the demonstrator rat, we let it eat rat chow that's been laced with cinnamon on the left-hand side. And then another subset of the rats, we'd let them come out. We then let them eat rat chow mixed with Hershey's cocoa powder. Yeah? And then we let them go back and they get to cuddle with the demonstrator rat, who has not directly interacted with the cinnamon or with the chocolate yet, at least like not in the context of rat chow, but they just sort of smell each other and they have chemosensory cues and they cuddle together, blah, blah, blah. All right, now we take that observer rat. Sorry about the police outside. We're in Tampa. And anyway, so the observer rat, sorry, maybe it's an ambulance. I'm sorry, I get distracted acoustically. Anyway, so the demonstrator rat then interacts with the observer rat. Then we take the observer rat and we let it choose, go out into a stage scenario where it gets to choose between rat chow that's been flavored with cinnamon and rat chow that's been flavored with Hershey powder. What do you think it chooses? Well, under most circumstances, it chooses the scented food that the demonstrator rat just interacted with. And then you can take this new observer rat and then let it be the demonstrator for another novel rat. And so you can create chains of culture that just based on an early chance thing that a demonstrator did, you either gave this original demonstrator rat a preference for an interaction with cocoa powder or cinnamon. That interaction, which wasn't even a choice for that first demonstrator rat, now results in a preference of the observer rat that can be transmitted to the next observer rat and down a little dynasty of rats with under experimental conditions that have long-term and stable preferences for certain elements of their environment. Interestingly, in rats, you can't get them to avoid food. So like if you take, so like using these dynastic sort of dynamics. So if I take a demonstrator rat and I give it rat chow laced with cinnamon and laced with, I don't know, ipecac, and then let it go back with the observer rat and it's like vomiting up its guts. The observer rat, if you take it out and you then let it choose between cinnamon and cocoa powder, even though its friend that smelled, or like its demonstrator rat, that's, I mean, I guess rats have friends, whatever. Anyway, so its demonstrator rat had interacted with cinnamon, had eaten rat chow, and was now vomiting. What do you think that observer rat does? The observer rat goes to the cinnamon flavored rat chow. So they can't actually link a negative outcome to the demonstrator, that it's ill. They just sense that their colony mate has eaten something, that it smells like this, and so they are primed to prefer that under subsequent choice encounters. Interestingly, if you kill the demonstrator rat, so if you just take a demonstrator rat and you sever its skull from its spine, and then roll the dead rat in rat chow and then cinnamon, and then take that dead rat and then put it in front of the demonstrator, in front of the observer rat. The observer rat does not have a preference for the cinnamon. All right, so a dead rat is not a very good demonstrator. So maybe that says that under extreme circumstances, the observers know to avoid the cinnamon, right? They have no preference in the next stage, although they have an equal probability of selecting cinnamon rat chow or Hershey's rat chow. But if the demonstrator, so that suggests that maybe the observers can tell if it killed it, then maybe you shouldn't prefer it. Okay, but then another neat study that they did, that Galef did, was that if you take a demonstrator rat and you pharmacologically cause it to sleep, you basically put it into like a little, not a rat coma, but like I guess a medically induced coma. All right, so a medically induced coma demonstrator rat, and you just smear some rat chow on its face with a little bit of Hershey's, and now it's asleep but not dead. And then you put it in front of the observer rat, and you just let the observer rat smell it, and then you let the observer rat go out and make a choice between cinnamon and cocoa. It will prefer the cinnamon. So even though the rat in front of it, the demonstrator rat, is asleep now and unresponsive, but smells like cinnamon, now the observer goes out and eats that thing. But if it were a dead observer, the observer, or dead demonstrator, the observer would have no interest in that flavor. Turns out in the end, it's rat breath. So observer rats will copy the foraging preferences of demonstrators so long as the demonstrator is still breathing. If it's not breathing, they don't attend to the cultural information, the recent foraging experiences of the demonstrator. But so long as it's still breathing, they copy it with very high fidelity. And so you can get transmission. Mothers produce offspring that have certain kinds of foraging preferences for, say, cinnamon, that become dynastic across generations under laboratory environments. And it has nothing to do with a gene that tells these mothers or tells these offspring to consume the cinnamon. It's just culturally transmitted. They know that this is successful, and they tend to copy the choices of living, at least still breathing, individuals around them. And for many species, those will tend to be family members. So families will resemble each other in their behavioral traits, but oftentimes they have nothing to do with the genes, or they have little to do with the genes. And I'll leave it there for now, and I will upload this video. It appears that this is going to be a 3 lecture, which makes sense. That's originally what I estimated it would be. Anyway, so I'll see you guys maybe, well, I'm recording this on Wednesday. I will probably see you guys on Friday, a subset of you. Anyway, come to my office hours because I would like to see, I'd like to see students this semester. I did become a professor so that I could teach people, real people, and not just talk to my lame camera. Anyway, so I'll talk to you guys soon. Take it easy. Oh, no, I'm not gonna look at that right now. Thank you though. And I will stop my recording.